This video will focus on the basics of DNA replication. Replicating DNA is essential for both reproduction and multicellularity. At the most basic level, for DNA replication to be a successful process, it needs to make an exact copy of the genome. Additional requirements include doing this with efficiency, not making many mistakes while copying DNA, and doing it at the appropriate time in the cell cycle. Our genetic material is brilliantly set up for this process, because making an exact copy of the DNA simply requires following the A to T and C to G rule, also known as Chargaff's rule. So, to make a copy of each strand, we simply need to separate them and then bind the complementary base. In essence, each individual strand serves as a template for the other. In eukaryotes, this involves multiple origins of replication that slowly spread and in the end replicate the whole chromosome. The newly synthesized strands are called daughter strands, and the original ones are the parental strands. So if we zoom in on one of the origins of replication, they look something like this. These bubbles in the DNA strands spread in both directions, but in the zoomed in figure here, we are only seeing one side of it. The continually unzipping DNA region is known as the replication fork. Helicase is the enzyme that unzips the double helix, and single-stranded binding proteins prevent the re-zipping of the helix. DNA polymerase III is our main workhorse enzyme, and it synthesizes new DNA. That is to say, it makes the daughter strands. We see it active on both of our strands here, so it is working on both sides of the replication fork to synthesize new DNA. However, this enzyme only works in one direction, so it is synthesizing DNA in the opposite directions for each strand, since these original parental strands were bound in an anti-parallel fashion. The two strands are called the leading strand and the lagging strand. The leading strand is synthesized towards the replication fork, and the lagging strand is synthesized away from the replication fork. DNA synthesis on the leading strand is straightforward and involves continuous synthesis of daughter strands by DNA polymerase III as the replication fork continues to open up. The lagging strand is a bit more complicated and consists of at least three additional enzymes. The lagging strand has to be continuously reprimed so the DNA polymerase can be attached close to the expanding replication fork. This is done by the enzyme primase. During this repriming and synthesis of DNA by DNA polymerase III, short fragments of DNA called Okazaki fragments are formed, and these must then be glued together. This is where our additional enzymes come in. DNA polymerase I excises the RNA primers and fills in the daughter strand gaps. DNA ligase then comes in and links the backbones of the different fragments on the lagging strand of DNA. If we zoom out slightly here, we see that this process proceeds in both directions along the DNA strand, and eventually these bubbles of replication connect with one another to form our replicated chromosome. I'll close this video by saying DNA replication is of course a bit more complicated than what is presented here. From what we know about this process, DNA is replicated in replication complexes pictured here. These complexes are set up to better coordinate the actions of enzymes like helicase and primase. It also is involved in associating two DNA polymerase III enzymes to replicate both the leading and the lagging strands. We will not dig into these complexes further, but I wanted to simply give you a sense of the complexity of this process. In upcoming videos, we will investigate why we have these differences in leading and lagging strand replication.